Hello, and welcome to the second part of Inside the Eukaryotic Cell. Today we're going to be focusing on these last three ideas, specifically which organelles are involved in protein production. We're going to look at what are vesicles and vacuoles, and then how does the cell get its energy. So to begin with, we're going to look at protein processing. In the last video we talked briefly about this, but some proteins that a cell makes are actually needed outside of the cell that makes them. So in order to get the proteins outside of the cell, they have to be packaged in what we call a vesicle. And a vesicle is a small membrane bound sac that encloses the proteins, and it keeps them separate from the rest of the cytoplasm. Now we have the endoplasmic reticulum and the Golgi apparatus. These are two organelles that are going to help in preparing the proteins to be exported outside of the cell. And we're going to talk about this in more detail here in a minute. So first off, we have the endoplasmic reticulum, sometimes referred to as ER. Okay, now this is an extensive system of internal membranes that's going to move the protein and other substances throughout the cell. The membranes of the endoplasmic reticulum are connected to the outer membrane of the nuclear envelope, and it's divided into two different portions. We have what we call the rough ER and the smooth ER. Okay, and their names are, they indicate exactly what it means. So the rough ER is kind of bumpy, and the smooth ER is nice and flat. So if you take a quick look here, the rough endoplasmic reticulum, or rough ER, this is where the proteins are made by ribosomes on the rough ER, which package the proteins into vesicles, and then the vesicles are going to transport the newly made proteins from the rough ER into the Golgi apparatus. And you can see kind of right here, like these little dots, this is representing the rough ER. When we have smooth ER, we are kind of missing some of those little dots or those little bumps. Okay, so the name rough ER, okay, means that there's ribosomes attached to the endoplasmic reticulum, and it's called rough ER because when you look in a microscope, okay, you can actually see that it has a rough looking appearance. When there is no ribosomes attached, this is the smooth ER because it has that smooth appearance or that nice flat surface. So what you need to remember is that the ribosomes on the rough ER make proteins that will be packaged into vesicles. And then the enzymes of the smooth ER are going to make lipids and break down toxic substances. The next organelle that we're going to look at is what we call the Golgi apparatus. Okay, and this is a set of flattened membrane-bound sacs. And what the Golgi apparatus does is it helps modify, sort, and package cell products for distribution. So when you look at the Golgi apparatus, usually you can see it's got kind of these flat pocket-like regions in it. Okay, this is how you can tell right away that it's a Golgi apparatus. And again, what's going to happen is that the vesicle is going to enter one side of the Golgi apparatus, and it's going to move through the Golgi apparatus, and as it moves through, it's going to be repackaged into a new vesicle. And it's that new vesicle that's going to be moved to the cell membrane so that it can be transported outside of the cell. So if you think about it, the Golgi apparatus is kind of like the center where things get packaged up and ready to go to kind of like leave. So it's kind of like a packaging center, if you want to think about it in sort of production terms. So when we look at the making and exporting of proteins, the ribosomes that are located on the rough ER will make the proteins, okay, which then cross into the membranes of the ER. The ER membrane then is going to pinch off and form a vesicle around that protein. From here, once that vesicle surrounded that protein, that vesicle then is going to go through the Golgi apparatus, and it's going to be modified by enzymes and repackaged into a new vesicle so that it can be transported later on. And the new vesicles then are going to be modified so that they can be released outside of the cell. So here you can see where that vesicle, okay, after it's left the Golgi apparatus, the vesicle is going to attach itself to the cell membrane. Okay, you can kind of see that right here. But if we look at kind of a enlarged view, what's going to happen is as that vesicle attaches itself to the cell membrane, it's going to kind of open up to the outside of the cell, and it's going to allow those proteins then to exit the vesicle. The next thing we're going to look at are what we call lysosomes. Lysosomes are vesicles that help maintain the homeostasis by storing and releasing a variety of substances as the cell needs them. Okay, so a lysosome is another vesicle that's produced by the Golgi apparatus. It's going to contain enzymes that are going to help break down large molecules. And then the lysosomes are going to recycle old or damaged organelles and digest food particles to provide the nutrients for the cell. Okay, we also have an organelle called a vacuole. A vacuole is a fluid-filled vesicle found in the cytoplasm of many plant cells, okay? Again, this is specific to plant cells only, not animal cells. Plant cells are going to contain a large compartment called the central vacuole, and it's here that the water, ions, nutrients, and waste is going to be stored. Once that vacuole is filled with water, the cell is going to become very rigid and it's going to allow the plant to stand up. When the vacuole loses water, the cell is going to shrink and the plant wilts. So if you think about a plant that hasn't had water in a couple days, or like, and in my head I think of like a beet field in the summertime or something when we are kind of going through like a dry spell, okay, what happens is the plant's going to use that water, and as it uses the water, it kind of depletes those cells of the water, therefore it's going to be kind of like a limp, or it's just going to kind of lay there. Okay, as those cells fill back up, it's going to allow that plant to stand back up. Now we have other types of vacuoles as well. Some protists have contractile vacuoles, and these are used to pump excess water out of the cell in order to control the concentrations of salts and other substances. We also have what we call food vacuoles. Okay, this is formed when the cell membrane surrounds food particles outside of the cells and it wants to move it inside of the cell. And the last thing that we're going to talk about in this section is energy production. So cells need a constant source of energy. The energy for cellular functions is going to be produced by chemical reactions that occur in the mitochondria and the chloroplast. Both organelles are going to have a chemical reaction that produce what we call adenosine triphosphate, or you will start seeing this written down as ATP. And this is a form of energy that fuels almost all cell processes. So this is something that if you go into the medical field, you will talk about a tremendous amount. ATP is very important. In plants, we have what we call chloroplasts, and this is an organelle that's found in plants and algae, and it uses light energy to make the carbs from carbon dioxide and water. Chloroplasts are going to be surrounded by two membranes, and they're going to have several stacks of flattened sacs where energy production can take place. And a plant cell may actually have several different chloroplasts throughout it. We will actually talk more about chloroplasts in another section, and we'll go into more detail in there. Okay, and some of you may have heard this before too, but if we talk about the mitochondria is typically known as the powerhouse of the cell. Okay, and this is, the mitochondria is an organelle that use energy from organic compounds to make ATP that the cell can utilize. So most of the ATP that's needed by a cell is produced inside of the mitochondria. This is something that is common to both plant and animals. The mitochondrion is actually surrounded by a smooth outer membrane and a folded inner membrane. So ATP is produced by enzymes on the folds of the inner membrane. So if you take a look right here, you can see that we have our mitochondria here. We have our inner membrane, okay, which is kind of like this shell here. This is our outer membrane, and then our inner membrane is like this little folded region right here. If you look at like a big picture of a cell, like what we have over here, the mitochondria is right here. And you can see that there's another one that looks like right there. And this is going to wrap up or conclude our video on eukaryotic cells for section 7.2. Please let me know if you have any questions. Otherwise, have a great day, and I will see you in class.